It is 11 a.m., so we're going to go ahead and get started. My name is Caitlin Wilkerson. I am a PhD student here at the University of Louisville, and I have the opportunity today to introduce our speaker, Dr. Ted Smith. Dr. Smith is the director at the Center for Healthy Air, Water, and Soil at the Christina Lee Brown Environmental Institute at our very own University of Louisville School of Medicine. And Dr. Smith's expertise is in community-engaged environmental technologies that provide place-based clinical insight for preventing or reducing the burden of disease. He received his BS in biology and psychology from Allegheny College in Pennsylvania, his MS and PhD in experimental biology from Miami University, and he completed his postdoctoral studies at the prestigious Massachusetts Institute of Technology. In his administrative capacity, Dr. Smith holds leadership roles in the Community Engagement Corps for UofL Superfund Research Center and the Center for Integrative Environmental Health Sciences. He is co-founder of the Christina Lee Brown Environmental Institute and serves as its liaison with government, industry, and philanthropy. Dr. Smith was previously the Chief of Civic Innovation for the City of Louisville, where he created the largest real-time asthma monitoring project, Air Louisville. Dr. Smith has secured over $10 million in funding for wastewater research activities from federal, state, local, and charitable sources, and authored over 40 peer-reviewed publications since 2021 covering his wastewater research endeavors. And without further ado, I won't take up any more of his time, and we'll let him tell you all about it.
All right, you got to love AI. So it wasn't hard to get a cardinal on a toilet or the thinker on a toilet. That took less than 20 seconds in both cases. So I encourage you to play with the technology. It's fun. So thank you for having me back. I think, I don't know, hopefully not like pandemics. My presentation recurs. But this time I wanted to do something a little different. Rather than focus on kind of trying to introduce wastewater-based epidemiology and how, what it is and how it works and all that. We're sort of at a point now, many years into this work, where I think we can maybe be a little more reflective about the work. And really, I took this as an opportunity to shamelessly use all of you to help me and others think about really what we should do next. And you can't do that without knowing a little bit about what we've done. So I'm going to try to breeze through some of that and leave us with some big questions at the end. So that's the plan for today. We are trying to get a health signal from garbage. And I've been using this slide since 2019. It's important that we understand, while it can be fun to talk about using wastewater as a research platform, there are lots of people, in fact, most people I interact with in the science community do think it's garbage, and they wonder why you would be working in garbage. And so, that's our humility, right? We want to think of it as, a clinical specimen, but it really looks like that always and everywhere. And so I think with our humility, we'll say, well, we do have some tools that can pull a signal out of noise. And this is probably the worst case scenario, right? I mean, people that have done air monitoring work, water work. I mean, that can be difficult. I think this is extra difficult. And it really is because the chemistry itself is, nobody understands it, right? I mean, nobody's actually spent the time to actually interrogate the matrix in all the major cities of the world or in septic tanks or whatever. It's just not been part of the science platform, really, until the COVID-19 pandemic. So we have to respect that. We have to understand that even the ethics aren't completely worked out. Every time we go to our IRB, they say it's not human subjects research, it's exempt. We have to keep going back every time we do something new, right? Because we're always finding something new to do. And they always come back and say it's exempt. But exempt is a technical term, related to very specific regulations around human subject research. On the ground in communities, what the IRB thinks isn't necessarily what everybody thinks, and so, in no small part, thanks to the pandemic, we've had a polarization in this country that is really unprecedented. And this kind of technology runs right into the middle of that polarization. So, for example, when we started the work in COVID, we were doing Jefferson County and Kenton County in northern Kentucky, right next to Cincinnati. And we had a good, productive relationship with Kenton County for, I'll call it 6 months. And 6 months in, the person that runs the wastewater utility was under so much pressure from the local utility board that they had to stop. And it was really unfortunate because we had really good data that was tracking well with what was going on in the health system up there, St. Mary, Louisville, whatever it is there. I mean, they were looking for any kind of information. The public health folks were, to understand what they would see in the emergency room. And yet, there's a little more about called a libertarian ethos, where they said, do not look at my feces and urine, that you're violating my privacy. So you should just know that that conversation is alive and well everywhere all the time, regardless of what our IRB says. So we're going to keep track of that. It's important. It'll continue to be important until, as a country, we reach some kind of resolution to this issue. Just remind everybody, one of my other favorite slides, kind of how a sewer works. I don't expect anybody to know this. I didn't know it until 2019. But I mean, it's essentially in most cities in America, it's a gravity-based system where, sewage is trying to run downhill all the time. And when it runs out of downhill, we pump it uphill and then it runs downhill again until it eventually ends up at a wastewater treatment plant. And in many parts of our community, it's a very long journey. It can take a day for the sewage to get from one place, a source, to the treatment center, really depending on which infrastructure it has to traverse to get there. So, of course, as scientists, we're going to want to have to keep track of time as one of those important variables, right? So we're not, unlike air pollution, where I measure what's in the air, that's what was in the air right then and there, right? I pull something out of the sewer, I'm not really sure when that was introduced into the sewer. And so there's going to be this range of possibilities. And then there's weird physical problems, like on hot days, the sewer is hotter than on cold days. And so the chemistry of a changing matrix like that is something that we don't fully understand. Nobody's actually studied it. So, I mean, we're several thousand papers into wastewater-based epidemiology now. Nobody's studied that question. So it remains one of these unknown risks for us. How does the matrix change? And then whatever I'm measuring, how might I misinterpret what I'm measuring? Some factor like travel in a sewer pipe. So there are countries that have much more sophisticated infrastructure. Israel has a very sophisticated wastewater infrastructure. They've got telematics through the line. And so they know not only the things they need to know, like flow rate, temperature, and all that kind of stuff, but they know some basics of chemistry of what's going on in their infrastructure. So it's possible. I guess there are Chinese cities that are trying to do very similar things right now. I mean, it's possible to do more. We all have come to hate our sewage utilities in this country. I'm not really sure. I guess it feels like the worst way to spend your tax dollar. It's like throwing my money away, in the sewer. And so if you look at MSD here, they're hated, generally speaking. I'm the board chair of TARP right now. I am usually grateful for MSD because they've got like $100 million problem and I've got a $30 million problem. So that's where we are in this country, aging infrastructure. So it's not a great place to start here. Is another sort of happy thing to share with you today. But it is an infrastructure that we learn how to work with. Okay, so how do you get sewage for sampling? Sort of three main ways to do it. We can go low tech and cheap. Sort of worth what you pay for it is going to be the punchline there. So I can dip a bucket and open a manhole and dip a bucket in, and I can get something. And of course, you would guess that's not going to be very representative much of anything, but what was going by at that moment. So very few people in the science community do work like this. There's times when you may want to, when you might need to get quickly somewhere for something. Hospitals often have this as part of the overall framework for monitoring. So if there's a concern about something, it's a good way to get a very quick snapshot, which might be indicative for further, more comprehensive follow-up. Composite sampling, composite, as the name suggests, is bringing together over time, and there's many ways to do it, but time-based composite sampling is the most common. So I grab a little bit, maybe every 15 minutes for 24 hours. You can guess that'll be much more representative of the population that's connected to the sewer. So this is the most common method. It is the most common field method. So it's what we've done since the beginning of time. We have we own composite samplers, and we borrow NSD's composite samplers. They're kind of expensive, and during the pandemic, ISCO, the company that makes them, it ran out several times, and so that and freezers was like the hard part of the pandemic for me. Okay, so that's a thing. It's complicated though, because you got to have people go out, open manholes, install things. You know, if it's in the middle of the street, you're not going to be able to run it inlet without, you know, closing it to traffic. We've got all those kinds of problems. So hunting for manholes that you can put that thing near for 24 hours is also part of the work, but it's doable. And then the most common thing by far is actually just walking over to the wastewater treatment plant, and in Louisville, there's 5 of them. And I like to think of it as like a Guinness tap handle. I've really never seen it, but you know, you draw what's coming out of the composite sampler at the wastewater treatment plant, and it's very easy to get samples out of the wastewater treatment plant. They have to do it because the Environmental Protection Agency regulates the industry with the Clean Water Act, so they are constantly testing mostly the effluent, but they also test the influent, the water coming into the wastewater treatment plant. So the infrastructure is there. It's easy to get the samples. So the vast majority of research in wastewater-based epidemiology comes from that kind of work. Okay, and so, you know, there are lots of things we don't know about how to use this platform because it's so easy and sort of representative in a sense to go to a wastewater treatment. So the CDC's National Wastewater Surveillance System, which at 9 o'clock this morning still exists, is doing that. They're getting samples from wastewater treatment plants all over the country, and they're trying to keep track of respiratory viruses nationally, just using that platform. Okay. Today in Kentucky, we have a Kentucky wastewater surveillance system. It too just relies on wastewater coming into the wastewater treatment plants, and we are part of that. Almost part of that framework for the state. There is another part of the matrix that only the team at Stanford feels strongly about, and that is what's called primary solids. Solids, as the name suggests, is the solid material leaving your body and entering the sewer. Early in the pandemic, there was actually sort of a, like, I don't know, like an East Coast, West Coast kind of gangster thing going on where, you know, people are name calling, like, I think we're looking at wastewater, you should only be looking at solids. And the good news for all of us is that Stanford lost. There's still some high profile people out there still doing important work, but it is just a much more limited proposition to work with solids. You can only be at the wastewater treatment plant. The kinds of biological material and chemical material you find is really caught up deeply in only excrement. So what we find is working in wastewater gives us a view into both, because there's dissolved solids in wastewater that we settle back out. So anyway, so just know that in case somebody quizzes you, there's lots of ways to do sewage. Okay. Another important concept that we're constantly thinking about is, you know, what is the scope or scale of the work that you're doing? And I do think we like to think that we could run backwards from a wastewater treatment plant and find a sick person. And that's been done a couple of times in the last few years with polio case in New York City, big city. Find it in a wastewater treatment plant that treats the sewage of millions of people, work your way back to an apartment complex, to an apartment unit. So it can be done. It's super expensive and, you know, kind of a gamble if you think about it. But that's really not what happens out. So back to the ethics issues and concerns about privacy, nobody is really ever chasing anything anywhere to that level of detail, because the cost just goes up dramatically as we move from scale to scale to scale. Most of our work at the University of Louisville has been in the middle and in the upper right. So we've done a lot of work at the neighborhood level, where either a neighborhood or a group of neighborhoods, we found a catchment area that was isolated and we could regularly sample from, or the wastewater treatment plants. For 2 years, I led the surveillance of the Kentucky State prisons. There's 14 of those. It's a kind of a campus environment, so it's certainly smaller than a neighborhood. Very important. It's a vulnerable population. They don't have a choice. So understanding what their infectious disease risk is a pretty valuable question to have answers to. So Kentucky, Ohio, Oregon, a handful of states that I think did a really good job on prison surveillance that was integrated with the way they delivered healthcare, the way they delivered vaccination. So it's a tool that can be used at the campus or a campus level. So we think about stuff like that because it's important. You know, if you look at sewage, you really should be asking the question, whose sewage is this, right? Like, who does it represent? Then on the lab side, there's two paths you can go on. So look at it, now it's all open. We've spent many years looking for infectious diseases. And so I think everybody in this room knows that means you're going to be on some molecular biology platform, you know, PCRs, it still is the most common platform for working with these pathogens. Because we have such a great, you know, genomics and bioinformatics core, you know, with Melissa and Eric, we spent a lot of time trying to do much more complicated work on the molecular biology side. A lot of NGS, you know, work, you know, trying to find individual variants and where they were and all that. And it was a lot of fun. And then we started to go look for other things using the very same platform. So how many pathogens can we find if we do broad untargeted sequencing? And, you know, all good and fine, expensive. And, you know, it turns out the country wasn't really ready to go figure out what everything was. They were really concerned about COVID, really concerned about the flu, Mpox, RSV, and the conversation sort of, you know, leads from there. Even with bird flu around, still not in the standard panel, which is interesting. Okay, so that's the path we have the most experience in and the most publications in as a university. The other path, which I know you all know, is the chemistry path. And there, I mean, the reason that I think less work is done here, and even throughout the pandemic, because you could go hunting for things with a mass spectrometer, right? You could go look for proteins, whatever. Dr. Merchant and I had that conversation at the beginning of the pandemic. The cost and the time has always been an issue with that platform. And so the cost and the time, it really is represented in the literature. I mean, this is an order of magnitude less research done looking for chemicals than looking for pathogens. And I really just think it's a feasibility discussion more than anything. Though there are discipline conversations, like as we all know, infectious disease people are different than toxicologists. And so, you know, they don't go to the same meetings, they don't publish in the same places, they don't talk to each other that much. So that too is a problem here. And I think the ID people will really run the table for years and years and years in this area and have early sort of leadership and what to do with it. Okay, here at home, we have a humble little organization that I have the privilege to direct, the Center for Healthy Air, Water, and Soil. And this is really the core team that we leverage to go look at anything related to wastewater. And so my colleague, Dr. Rochelle Holm, in the lower left, she's actually what I call a field station for the University of Louisville. She's in Malawi. It's a long story how she got with us, but turns out many of these small middle-income countries have to do the scrappiest work, and a lot of that work can involve sanitation issues and infectious diseases. So, you know, these are people that will, I mean, Rochelle will tell you, working in pit latrines, and, you know, looking at open waterways and really trying to figure out what the hygiene is and what the infectious disease risk. So we learned a lot through the pandemic from these smaller countries, less developed countries, because they had to be more pragmatic about their work, and they were sort of innovative, right, in figuring out how to get signals out of noise. So Rochelle's been tremendous since she joined our team. And then Lauren Anderson, who has sort of served as a project leader, project manager for our various program projects, and Kyson Black, who has joined us about a year ago to lead our data analysis. The operation is really, how do we guide field work? How do we source lab work? And then how do we integrate data, and especially data with clinical data? And that's really the way we divide the work up. And then, of course, because we're in a wonderful university, we have the opportunity to then work with different entities, right? So CIEHS is an important partner for us as we make this shift from infectious disease to environmental toxicology issues. And so obviously the metabolomics, core, and all, I mean, the facilities to look at what pollutants can do are truly here at CIEHS. And then the ones that aren't are over at the Superfund Center. And so, we find ourselves increasingly working with these two centers and sometimes working with them together, which is awesome. We also have a handful of separate, completely independent initiatives, or maybe semi-independent. The Green Heart Project is an R1, but it has components that are aligned with the Superfund program. So on any given day, we can sort of squint. We can talk about it kind of in either column, but in its dedicated funding, there are a lot of opportunities where we look at wastewater, and we have been collecting wastewater in that study site for a long time, so that we can answer some other questions related to that greening intervention as part of the Green Heart. And then we recently got, and it still exists today, the EPA EJ G2G grant that APCD got for resurrecting the air toxic study in West Louisville. That project starts collecting air samples and wastewater next month-ish. It's been sliding a lot, and if they let it keep sliding, it could get canceled by this administration. So I'd really like to see it get started. But that's a great example of just leveraging this other infrastructure that we have into being a good community collaborator with a larger environmental justice initiative. All right, key statistics. So I think we're at about 49 publications since 2021, about $12 million in direct costs. And we have 5 -80 freezers full of sewage in Baxter 2, 3,354 wastewater samples, about 500 milliliters in each sample, about. So it's important to know that. I mean, we've been able to leverage the repository for lots of little pilot projects and go into what if questions. So just know that you've got a what if question, and it's sort of in the time period of 2021, 2022, 2023, and then going forward, we continue to accumulate. We only own 6 freezers, so there's probably a point in time when we'll stop accumulating. And then if you look at sort of the areas of expertise, no surprise, most of the stuff is infectious disease, most of the stuff is COVID. A lot of it is epi, so analyses to compare what we find in the wastewater to some kind of clinical data source. Which makes a great deal of sense. There are some lab methods papers, which is great, where we try to understand whether there are markers for population that we can use to normalize samples, or whether we use machine learning to figure out how to make flow rate calculations differently. So we've tried to do some stuff with the lab, but that's really not our strength. And quite frankly, if I look at the Stanford group, their strength is 100% the lab, 0 on health, 100% laboratory methods. A lot of people came into this field came in from doing environmental laboratory work. And it makes sense, right? Like who knew anything about wastewater? Environmental wastewater people knew about wastewater, and they were interested in the kinds of pathogens that are in wastewater and might be in surface water. And so they know the lab methods, and they're the innovators in that area. They don't know anything about health, generally speaking. And so the big bridge for most of the field was trying to get smart about being, about how to talk about the health implications of what you're finding in the wastewater. And that, I think, has been heavy lift for a lot of folks. We've got some public opinion work. We're the only group that has done a national public opinion survey on the ethics of privacy concerns around wastewater. We've run it twice for the national sample. Amazing. Nobody else cares about this, but we think it's important to stay on top of where the sentiment is in this country for this kind of work. And then really, where most of us came from, chronic diseases are the bread and butter of a lot of our operations. And yet it's probably where we have the least credibility at the moment for the wastewater work. And so we're really trying to leverage a lot of infectious disease work, a lot of working with this kind of sampling and this kind of data to address other kinds of problems. And that's going to be one of our great challenges. We're not going to read all these publications, but I wanted to give you a sense for, kind of the productivity. So for us, we really got serious and started putting resources against us in June of 2020. So you should expect a lag to see the scholarly work out of this. But 2021 was sort of a good sort of first kind of breakout year for the team's work. 2022, now we're getting a rhythm. It's all good. Some of these papers, I've just listed the ones that just have higher citations. So nobody's surprised that Nature or Lancet, might fare better for citations than some of the other journals, but that's of course not always the case. Every now and then there's a little journal that'll pop with the right thing in it. 2023. So, now we're sort of in these papers are like, let's call it a pandemic wrap up. And so some of these were bigger data sets that took a lot longer to put together. And then there's a fair amount of commentary sprinkled in here, trying to message to policymakers and others that we should be thinking about using tools like this more aggressively than we have been. And then 2024. So the mix of work that we're publishing now includes these new kinds of areas of exploration that we're going to sort of finish with today. And so aside from Rochelle's work in Malawi, which will always have infectious diseases in it because of the nature of her context, increasingly our work, will not be about infectious disease. We have a large contract from the state to go keep chasing respiratory viruses, but it feels like contract lab work. And so we always ask ourselves the question, how much longer do we want to do contract lab work? I mean, it's nice to get the money, but there's an opportunity cost, right? So we're thinking about that all the time. And then on the funding side, I think pandemic was pretty good to us. We were busy. We, I think, brought in and leveraged a lot of money across the university. I mean, at the core, of the pandemic, we probably had 25 people working at the university on stuff regularly. We were buying a freezer, every 6 months. So this money, the vast majority of it is COVID money. And so with humility, we can recognize that, well, that's a big number. That's a number we're not going to see again. And so, except for the Air Toxics grant, which we're a sub on, and nobody's getting rich on that grant, we're going to have to seriously think about where this fits into other funding mechanisms. Even the CDC, which funds wastewater research, is under pressure by CDC epidemiologists to not do this work. It's really interesting. Like when you try to do something new, it's a little bit counterintuitive, but the first resistance you meet is the people who know the most and are closest to you. And they will have a difficulty accepting it and endorsing the change. And we've met that everywhere we've ever gone. So at the state, at the federal government, if you're trying to do something new, it wasn't in their textbook when they got their degree, it's not a thing. And so it's a really hard challenge here to not have the whole science community, and especially the relevant science community, on board. And so the first two R01s that we put in this area, the study sections were brutal. That's garbage. That's not research, right? Like, that's not a thing. Nobody knows anything. It's witchcraft. Like, nobody knows how that works. Never send us something like that. And so this is just gotta realize it. Like, got enough scars. Like, all right, I get it. We have to hide it under something that you think is legitimate until I show you that it works, which is fine. But that's the situation we're in. And certainly with this administration, it's really a coin toss where this all goes. Ironically, the $8 million in total costs that we got from the CDC came from Trump 1 administration. The Undersecretary of Health and Human Services loved wastewater. And so we were in the right place at the right time and got that $8 million. And that really was an engine of a funding source. So it's not unimaginable that something good can happen. I don't know. I'd like to be optimistic on it. Samples, just so everybody knows, 3,354. Virtually all of them are from Jefferson County. Most of them are from sort of neighborhood scale sampling, which is great. And it's helped us with some of this other investigation work we've done where we've been able to pull out of very different neighborhoods. Some of them are industrial, some of them are completely residential. So the repository is proving its worth. I think the fact that it's a secret is a big problem. So one of the reasons I want to highlight it today is we have a repository. We keep the freezers running. Some people scratch their heads. Why do we have a repository? But we do. We've got some little niche stuff that we've gone chasing, but just to give you a sense, like there's other stuff that we might be able to just get lucky and we might have a sample that can give us some information about a place, but that's a little more limited. Okay, not going to go through all this. I think you all understand that there was a tremendously reliable correspondence between finding pathogens in wastewater and finding them in the clinic. And I don't think I've not met anybody that says that's not true. So here's the good news, it's 2025 and everybody understands that the prevalence of these infections for certain pathogens can be well and reliably tracked in wastewater. Getting folks to operationalize it, still a hard, heavy lift. But that's the work that we're doing. I cherry picked this because it's the best graph that I have for this work. And this is work we're doing with JCPS. So it took 2 years to get through their IRB. So anytime you complain about IRB, I want you to go to JCPS because you'll be scared. To get attendance data by school. And so 2 years later, we have attendance data by school. And so this is COVID, SARS-CoV-2 relationship for southeastern Jefferson County. Blue line is sewage. Yellow line is attendance. And so, like I said, I picked the best graph. The other ones, you might be a little more skeptical, but we are working through these lag analyses to try to figure out how we might be able to sit down with the school district and say, you know, based on what I'm seeing right now, for flu, COVID, or whatever, what I'm seeing right now, it does translate into attendance in next week, 2 weeks from now, 3 weeks from now. That's where we want to get with this work because they get paid for attendance, right? So this is something they care about, money. So if I can develop any kind of relationship that's even roughly right, it is probably useful for the school district. And our best partner is the chief medical officer of the school district, Eva Stone. She's tremendous. She's like, I want the wastewater data so that our nurses in our schools and that everybody else can understand how important it is at certain times of the year that we up our game on sanitation or we up our game on how we're dealing with large crowd assemblies or whatever it is. I'm really hopeful for this. Okay, leaving infectious disease. So believe it or not, before the pandemic, we met some folks in Arizona who were really trying to figure out the chemistry questions, you know, like how do I use sewage to find opiates? How do I use sewage to look at caffeine consumption patterns? Rob Halden is a wonderful person, truly a pioneer in the field, has more sewage in freezers than we do and called it the Global Wastewater Observatory, but it's a bunch of Best Buy, like regular 32-degree freezers. Anyway, in hallways at Arizona State University, which is hilarious. But it is the Global Observatory, and Rolf is a believer, and he's been doing it for a long time. We worked with his team to develop the first assays to go look for the urinary metabolites of exposures to a panel of air toxics, all organic compounds. And so the graph in the upper right, the plot in the upper right, is a bunch of those metabolites, and we did sort of a rough hack. You know, here's a community that has a lot of industry in it. Here's a community that doesn't. And do we see different exposures? Do we see different exposures as based on the amount of metabolites we recover in the wastewater or the types that we recovered? And, you know, it's not anything to write home about. You know, I think this work that we started with ASU was just sort of directional. Like, is this even something that we can do? We got into all kinds of issues with smokers, and this is not my expertise, but so like normalizing with cotinine so that we try to like dampen the effect of smoking turns out to be one of the harder problems that we have to work through here. That's why we look for these extreme places with high exposures, and then everywhere else, because the smokers aren't likely to overwhelm the signal in that case, right? And they're in the background everywhere, theoretically. So it's a challenge in this area. We've recently redone that work, sort of brought it to UofL out of ASU, and Pavel has been leading the charge. And, has both with targeted and untargeted analysis with assays we've developed, I think he's done an amazing job. Went to the repository. We have longitudinal data for a bunch of these places. And so what you're looking at is sort of 4-week data, serial data for, I don't know, 15 sites-ish across Jefferson County. And the story, depending on which metabolite you're looking for, is a lot more interesting than what we were looking at before. And so we're working through publishing this manuscript now, which is mostly a laboratory methods paper, which is great. We've established what we did, how we did it. And then the next work is really to do a better job than applying that assay to some very specific circumstances where we understand what the air pollution, what's measured in the air, or where we understand what's in the urine of people in that area, so that we can make sense of this data that really is hard to make sense of without something to triangulate with. I'm just sharing the fact, you probably all know this, that there's lots of folks, especially with the opiate settlement money, that are busy chasing opiates, fentanyl, whatever, in the wastewater. It can be done. There's not a tremendous amount of enthusiasm in the scientific community generally for this work. It's not really clear to me what most communities are doing with this. We, as a matter of policy, don't do this work because of the great history of stigmatizing neighborhoods in Louisville. So I think it's going to be hard for us to be a good actor, out there helping with air pollution issues when we're also busy telling the police and the public health people where all the opiates are. So it's tricky. So we just decided, as a matter of principle, we're going to let everybody else sort it out, and we'll catch up later on that. I'm going to pass through that. Some of these other chemicals, though, a little less controversial. There's a great history, with nicotine. It's well described in many cities across the world. It tracks well with smoking rates as measured by other sources. So, I mean, we can start thinking about leveraging a different part of the literature for things that are a little closer to home with us, whether it's substance use or other chronic condition issues. Thanks to Dr. State's leadership, we've been really trying to understand metals. We're very early on with that work, but there's not much of any literature on metals in wastewater. So it's a sort of a green field for us to go run into. It has any number of challenges in it, but we're mildly optimistic we can figure this out. Getting it funded is really going to be the thing. Right? So getting it published, we're almost there. Getting it published, then getting it funded. So that's where we are with metals, but it could be really promising, and it really makes sense in cities that have industrial areas or a history of industry, because we all suspect that, that gradient could be important, and it's very difficult to get it in other ways, right, in other methods. That work sort of led us to, with Dr. Dupre, some work on cancer clusters. And so we've looked at some of the cancer clusters that we have here in Jefferson County, and we looked at the metals associated with those clusters. That led us to bring in Kevin Sokolowski, one of my heroes. He said, I don't understand why we don't just use the Cologuard test on the sewage. Of course, when you say that out loud, it sounds silly, but it's not silly, because it's hard to detect colorectal cancer. And the guidelines say you have to be 50 before you start getting screened. And people in their 30s are developing this disease at high rates. Okay, so we have a problem in this country with colorectal cancer, and we know that detection is a really important part of reducing mortality, complex morbidity. So why don't we figure out how to screen for cancer, the cancer itself, not the risk factors for cancer? And so I found a company. Okay, so you can think, why not Cologuard? Because half of Cologuard's analysis is, who knows what they're looking for. Half of it's like a genetic marker, and what's the other half of what they're looking for? Colorectal cancer. What do you look for in the toilet? Blood. Sewers are full of blood. All right, happy Halloween. So guess what? Running Cologuard in the sewer is a waste of your time because everybody's got cancer according to Cologuard in the sewer. So we found a company in St. Louis called Genoscopy who just got approved for a genetic-only home screening test. And we found them at the right time. And the woman who runs their research is a graduate of WashU, and most of the science team there is too. And they said, we love the idea of testing sewage. And so we have a data sharing agreement with them. We sent them a bunch of sewage from the hotspots and matched controls. And, we're just publishing the sort of first take work on that. The column to pay attention to, let's see if I do this without advancing the slide, is this adjusted CDHI to GAPDH, which the GAPDH is like a housekeeping marker. For us, it's a lot like just trying to keep track of the population that's present. And so the ratio data, if you squint, we're feeling like we're seeing cancer in places where we know we see cancer. There's all kinds of problems with timing with cancer, right? So people had cancer, people that might not have cancer now, they're in the hotspot because they had cancer, not because they have cancer. So it's going to be a hard problem in this area. But generally speaking, if we could do population screening, relatively small neighborhoods, like a roving spotlight, right? Not picking on anybody for any particular reason. A roving spotlight, you could imagine directed follow-on screening campaigns into those neighborhoods. In Chicago, they've done that. Their public health department took voter registration data and socioeconomic data, and they found neighborhoods to do targeted Cologuard campaigns against, and they pulled out an unbelievable amount of cancer. So it's not crazy. And it would be helpful if you actually had a reason to be somewhere, not a statistical reason, but an actual biological marker reason. So fingers crossed, we're, I think, pretty excited about this kind of work. This fits well with the sort of SBIR, STTR framework. So the company's very happy to go pursue that with us. I'm not really sure, at some point in time we'll figure out that we don't have that much to contribute, but for now it's a good deal. We have tried to apply this stuff in disasters, and this is now an old slide. Back when we had a chemical hoarder here in Jefferson County, it was a great sort of natural experiment opportunity, a house full of chemicals. Somebody said, let's burn it. And so weirdly, so perversely, we got excitement. Somebody said, let's burn it. And so we said, well, gosh, this would be great because we can get the before and then they'll light on fire and then we'll get the after and we'll see what everybody was exposed to. But calmer heads prevailed and they decided to knock it over and clean it up instead of burning it. So we didn't get to do that experiment, but that kind of framework is alive and well for us. And every time something bad happens somewhere, I appreciate the phone calls that we get to say, well, why don't we go look at the sewage? And the reason we're not generally looking at the sewage is we don't know what the sewage was before, right? It's just a huge problem in this area. And I will lay that at the feet of the federal government and its states for not doing something very low cost, like banking, thank you, that's my warning, banking their normal sewage, right? Even the CDC, the National Wastewater Surveillance Program, throws out the sewage the week after they publish it. So we miss the opportunity to answer some questions because we can't show up after this stuff and really make that much sense of it. And this brings us up to today, actually to Monday. Thanks to Dr. K, Dr. States. We decided to be time sensitive about a time sensitive funding mechanism. And so this controlled burn that happened over New Year's in Calvert City, Kentucky, was the impetus to go pour resources together and some terrible emergency sarcoma cases, an opportunity for us to show that we can go and respond to a troubled area like Calvert City. And so there you see, we said, we're going to bring the whole circus to town. So we're going to go look at the air, and believe it or not, the state had already spent a year and a half doing air toxics monitoring very recently down there. So we could stand on the shoulders of good, robust air monitoring work that they had done, and we could sort of replicate it. They already figured out where the good sites were to monitor from. It was very helpful for us. We could layer in the wastewater work, and so weekly just tracking these metabolites in the wastewater. And then thanks to my colleague Luz, some important community engagement work, to really have the community, which actually is the reason we know about the problem, because they reached out to our team, that we're going to be a responsible partner, right? We're going to try to figure out how to be helpful with their biggest concern down there. We're at a community of 2,500 people with 5 petrochemical plants and a Superfund site. I mean, I think they could use some help, friends. So we will keep our fingers crossed on that R21. We've done a little bit of work. This is from the R01. The second R01 that got rejected, I think triage straight up, was work really trying to look at therapies for cardiovascular disease in the Green Heart neighborhood. And so, we worked through a nice framework with Andrew DeFilippis on how we would really understand cardiovascular risk and treatment using wastewater. Again, that didn't fare well under review, but we now are excited about chasing the pulmonary side of the equation, because there is a literature. This actually paper came out this week. There is a literature developing around looking at the treatment of asthma in places that have greenness, or don't have greenness. And we have a pediatric pulmonologist collaborator, Scott Bickle, who's been, he's got a sub-cohort down in Green Heart, a study area. So we feel like we could probably start working on this side of the therapeutics, chasing funding on that side, maybe work our way back to cardiovascular at some point in the future. Okay, we're at probably 5 minutes. So what y'all know, the hard problems, the ones at least that I think about a lot. To do this work, we have to work with utilities. These utilities don't have to work with us. There is no way you're going to get sewage in any city in this country without dealing with that utility, right? You're not allowed to go open a manhole and put up a tube there. Not allowed to do that. So we have to figure this out. And the case in Calvert City is a really good one, where we're hoping that the wastewater utility will come on board with us if this were to be funded, but it's not guaranteed. And again, it's under political pressure everywhere in this country. So you got that issue. Population adjustment's a huge problem that nobody wants to talk about. So if we don't really have a good handle on the number of people, unique people that are contributing to the sample. And we have hacks like using peppermint model virus, where we say, well, here's a tracking virus that's generally associated with consumption of food, with people. It is generally, it's an okay thing. It's not a good thing. We have yet to find a good population marker that the community accepts as good. As and the work to actually go do it at scale is very expensive. Right? Like, how could I know how many people were in the system, and then how could I look at my marker and be sure that it corresponds? It's a hard problem, but it has to be solved because it really is getting in the way of learning things. Signal sensitivity, we still don't really understand how different kinds of targets get diluted, and really what limits of detection are functionally. And so it will always plague this until we understand how the infrastructure works and how it corresponds with the actual underlying biology and chemistry. So hard. Matrix is full of garbage. So a lot of the issues we have with next generation sequencing were associated with other biota, right? That were having their own day today, making their own byproducts. And, we need to know a pathogen that's attacking a human, not a pathogen that's attacking a raccoon. And so, or a rat or whatever. So, there's a lot going on in there. It's a little overly simplistic to say, well, this is just a bunch of human waste. Not so hard. Combined sewers versus dedicated sanitary sewers. So Many sewer systems in the country also accept stormwater. And then that not only does create pollution, it also introduces other pollutants into the matrix. And then we want to be clear about whether we're studying rain or we're studying humans. So that's hard. Travel time we talked about, dilution. There's other sources of dilution other than stormwater. So when we're doing prisons work, we weren't taking samples on laundry day. Big shock, right? Guess what? Laundry day, it's nothing but laundry water. Nothing but laundry water. And a prison, there's a lot of laundry water. So, it wasn't Monday ever, right? Thinking about stuff like that isn't very typical, right? Data about the households themselves, most of the papers that I've had the opportunity to review, the weakness is always on the clinical side, always. Bad data, poor data, sloppy data about the people, and a lot of great data about the laboratory methods. And so until we have a good handle on who's above ground contributing to this matrix, it's going to be hard to make big breakthroughs. I mean, think about communities that have big tourist populations that move through. I mean, San Diego, I've worked with early in the pandemic. On the weekend in San Diego, it's a whole bunch of other people that don't live there, right? And so they weren't learning anything looking at weekend data at all, because a couple million people showed up and that they don't know about and left, right? And so, we have to think about, it's not just clean, it's like people sitting at their homes on their toilets. That's not how it works. People are moving around all the time. So we have to acknowledge that. National repository we mentioned, it's a huge issue. I think I don't know that this administration is going to tackle it, but somebody should just, it's not that much money. Call it $100 million. $100 million, we get a big national repository, and we actually can ask questions, lots of questions over time. When did the pathogen show up in our borders? Is there a bioterrorist event happening over here? These are things we get answered if we had a repository nationally. The stuff doesn't cost that much money. All right, so I'm gonna just tell you, these are the directions that we are thinking about and in part doing work in. I would love, and we can take it up in part 2 after the high school team shows up at the RIG meeting, and then have a discussion about it. But I just want you to know this is generally what our team is wrestling through right now. We don't have a bright future in infectious disease work. So we're going to have to think about where we're going that fits with all these other research activities we're doing here at the University. So I will take questions if so.
We have time for about one question.
One good question. Who has a good question?
You made some great progress in papers, and you really talked about the spectrum of ethics and laboratory methods. What do you envision happening to help let the community in on this? So it's very population focused. You're worried about ethics. So how do you bring the community into the conversation, like through op-eds or something like that, so they can be in on this? A lot of science and technology comes over us away, and the community is left behind. So how are they factored in and how you grow this?
It's a great question. I don't have an answer for it other than to say we're trying to also manage being responsible, and part of responsibility is communicating. I get that, but part of it is not working with incomplete information as if it's complete. And this has been a big concern that we've had, is getting ahead of what the science tells us about things. I like to describe the work as we're trying to figure things out, and if they work out, it would be great to work together to figure out how to best use it in this community. But it's early, and I think we just tend to be a little more conservative about early science, right? That isn't definitive, that we don't know exactly what it means, but it could be promising. My experiences of working in communities is there's such a hunger to get to solutions that sometimes it can create pressure to use the information when it's not quite there yet. I like to use this is aging me, the Scooby-Doo reps, like we're just a bunch of meddling kids, right? That's what we're doing. When we went to the state fair and we said, we're gonna look for bird flu, we got everybody in the state upset. We got the Department of Public Health upset. We got the ag department, everybody got really upset with us. And it is because they wanted to know if they'd have to shut down the state fair. They wanted to know if they'd have to contact trace 150,000 people. Right? I mean, like, I'm sitting on a call, it's like, no, we were just going to look at the sewage in the dairy barn, right? And it turned into an unbelievable mess, right? To the point where it got shut down. So now we don't know anything about the dairy cattle in Kentucky. Well, we could have known maybe something, but we, didn't want to overpromise what we were doing. And there were people that were just ready to say, what are we doing with your information? I was like, well, I don't know if the information's that good. Well, we have to assume it's good. That isn't when we start. Usually it's bad. And so anyway, so I think this is going to be the tension, and I'm open to help with getting comfortable with those community conversations, but that's really been our history with this.
100% agree with everything you said. And I would only put it out there for the sake of discussion that we as a science community have a history we hear journalistic. And so the spot of knowing when to pivot into bringing them into the conversation, I don't think that's.
Thank you.